Okay, well, welcome to your Thursday afternoon, everybody. I'm thrilled to introduce the speaker today. I'm Neil Kelleher. I'm the director of the CLP Institute here at Northwestern. And today we're thrilled to have a relatively new, but although not becoming very new, I think many of you know her. And we're welcoming her to a chalk talk here, something we do as a tradition in the Institute. And the chalk talk this year will be with Shana Kelley, and the topic of her talk are rare cell profiling for therapeutic discovery and development. Some really exciting new platform aspects that you'll be introduced to. And for those of you who are familiar with Chemistry Life Processes Institute, it's a nexus for biomedical discovery at Northwestern. More than 65 faculty members, 67 in fact, across both campuses, belong to the Institute and collaborate across disciplines to develop new drug therapies, diagnostics, and other technologies. We also provide training for next generation interdisciplinary scientists. A bit about Shana here. She joined the faculty in fall of 2021 as the Neena B. Schwartz Professor of Chemistry and in the BME department. She's an extraordinary scholar and pioneer in translational bioanalytical research, something that's near and dear to my heart. And she uses a truly multidisciplinary approach to develop new tools for the diagnosis of disease and development of new therapies. She's listed as a top 100 innovator in MIT's Technology Review. She holds over 50 patents, founded 3 diagnostic companies, 2 of which have been acquired with a successful exit. She's received many awards and distinctions, ACS Inorganic Nanoscience Award. She's a Sloan Fellow, Fellow of the American Institute for Biological and Medical Engineers, and the Royal Society of Canada. Her education took her across the country, and she got a BA from Seton Hall and a PhD from Caltech. She completed an NIH postdoc at Scripps, and then started at Boston College, moved up to University of Toronto, so UT, where she last held the position of university professor. And we are absolutely ecstatic to have her as one of the new cohort of Wildcats at Northwestern. And just a little bit of housekeeping: keep your microphones turned off throughout the presentation to suppress unintended interference. And then after Shana's presentation, there'll be a 15-minute Q&A, and then feel free to turn on your video and microphone to ask questions and interact. You can also hit up the chat. The chat can be very active there, and we'll record to share it with those unable to tune in. So without any further ado, I give you Shana Kelley.
All right, thank you so much for that introduction, Neil. Very, very kind. I'm delighted to be here today. I actually panicked when I was first asked to give a chalk talk, because you'll see from what I'm talking about, it's very difficult to draw the kinds of structures that we work with. But I was very relieved when I was told that I could just use PowerPoint and kind of, PowerPoint you to death with my science. But anyway, that's what we're doing here today. I'm very happy to have this chance to tell you about our work. We work, as Neil said, we're very multidisciplinary. We really develop new technology platforms and then deploy them towards interesting unmet needs, biomedical problems, applications that really need new solutions. We do a lot of work around sensor development and detection technologies. We also have a platform for molecular delivery that we've done a lot of work with over the years. But today I'm going to focus in on our work on single cell and rare cell profiling. Tell you a bit about a platform that we've developed recently and a variety of different applications that we are deploying it towards. And the idea behind the work that I'm going to tell you about is that we have benefited so much from the ability to look at single cells, to take very complicated biological systems and really dissect them a single cell at a time and to collect that level of information. And there's a lot of powerful techniques out there to do that. But what remains very difficult is to look at a large number of cells at the single cell level. It's easy to look at 1,000 or maybe 10,000. And then when you get up into the hundreds of thousands and millions and up to billions, it becomes very difficult because if you're looking at them serially, it takes a lot of time to make those measurements. The other thing that's very challenging is looking at really heterogeneous mixtures of cells. So it's again, pretty straightforward when you have a single cell type and you're looking at maybe epigenetic drift, that kind of thing in similar cells, but more complicated when you have very different types of cells all in one sample and you want to collect single cell level information. So that was a type of capability that we were hoping to develop. And I'll tell you about some of the development work that we did on a new platform, and then take you into a few different application areas where we have used this capability to hone in on tumor cells in the blood to really be able to study them for the management of cancer. I'll tell you about some work we've done trying to look at very rare T cells in the blood to better characterize them for disease screening. And I'll also touch on some work on looking at CRISPR edited cells. And so I'm going to kind of take you on a survey of a few things, but I'll spend maybe half the talk in an area related to cell therapy development. I think that's what's in the abstract, and I'll stick to that. So I'll get to that topic in a little bit. The technology platform that we developed to address this need is called magnetic ranking cytometry. And this is an approach, you may be familiar with flow cytometry, where you take a fluorescently labeled antibody and you throw that into a cell mixture. You process the cells using a flow cytometer. The cytometer then reads out the amount of fluorescence on each cell and gives you a measure of protein expression. Here, instead of fluorescently labeled antibodies, we use magnetically labeled antibodies. Antibodies labeled with a very small magnetic nanoparticle. These nanoparticles can stoichiometrically load cells of interest according to levels of protein expression. So the antibody is specifically binding to a protein on the surface of the cell or even inside of the cell. And so we take a collection of cells, bring the magnetic nanoparticles in, and then the way that we are able to profile these cells at the single cell level is using microfluidic devices that will separate the cells according to levels of magnetic labeling. So again, instead of measuring fluorescence, we're counting magnetic nanoparticles and doing that using microfluidics. And so one of the devices, let me go back. One of the devices that we developed is shown here. And if you look carefully at this microfluidic channel, it's quite shallow. So when the cells come into the device, the channel's pretty shallow. And then as the channel keeps going, it gets deeper and deeper. What that does is it actually creates a velocity gradient. And that velocity gradient allows you to separate cells with different numbers of nanoparticles. There's magnets above and below the channel. And as the cells travel through the channel, they're responding to that velocity gradient. Cells with high numbers of magnetic nanoparticles will stop in the early zones of the device. Ones that have fewer numbers of nanoparticles will keep going and settle in the later zones of the device. So if we want to do an analytical measurement using this approach, we process the cell mixture, and then we come in with a cocktail of antibodies, other types of chemical stains. We visualize the cells on the microfluidic array, and then we generate the kind of histogram that you see on the bottom, where we're looking cell by cell. So collecting that information at the single cell level, and then having this histogram that allows us to say something about overall levels of protein expression in this mixture, as well as the single cell level information. So just to show you some data that we can collect from this type of approach, here we're looking at cells, we're profiling them according to levels of EpCAM. So this is a surface protein that you find on cancer cells. And here we have 4 different cell lines with 4 different levels of EpCAM. You can see that from the flow cytometry traces up top. And so you can see how we're able to tabulate this information about this collection of cells and quantitate protein expression. Same way you can with flow cytometry, but with very, very low levels of cells. We can also look at multiple markers. We can take a sample, we can divide it up into aliquots, and then be able to quantitate different proteins of interest. And what's very powerful about this is that even if we have just, let's say, 10 cells in a milliliter of blood, where there are billions of normal blood cells, we get a very consistent profile, whether we have 10 cells or 20 cells or 40 cells, or if we had 1,000 cells, that protein expression measurement stays very constant. So that's what this platform can do that a flow cytometer or other types of single cell technologies can't do. And we've shown that this works with surface proteins. So there, the magnetic nanoparticles and the antibody are being directed to a surface protein of interest. We've also come up with ways to actually fix and permeabilize the cells and look at RNA markers or also intracellular proteins. So we've built out a number of different assays for this technology platform. And then we've also learned how to make it preparative. So in the device that I showed you a few minutes ago, we're basically arraying the cells along the channel. We fix them, we permeabilize them to stain them, and they're difficult to recover. In the device shown here, we actually create different compartments along the fluidic channel. And then we trap the high expressors, the low expressors in separate compartments. We can remove that compartment and these cells can be expanded. They're very viable and they can be sequenced, they can be characterized. You can do all kinds of offline analyses. And what's very differentiated about this approach is that you can process billions of cells an hour. So it's very fast. We can flow cells through here with a pretty good flow rate. And so we're able to get levels of throughput and processing that aren't possible with things like a fax machine or other types of cytometry. So this platform, both the analytical capability as well as the preparative capability, is something that we've deployed looking at many, many different problems. I'm going to just touch on a couple of areas and then go deeper into another. We've done a little bit of work on using this approach for liquid biopsy. So looking at rare cells in the blood to avoid having to biopsy a tumor, but instead just looking at bloodborne cells. We've used a related technology to drive studies where we're using functional genomics for therapeutic target discovery. We've also taken that same approach in vivo and done screens in living animals. And then I'll spend quite a bit of time talking about cell therapy development, which is another great application for this platform. But I'll start just with a couple comments about what we've been able to do related to liquid biopsy. So as I mentioned, you can take a whole blood sample, take your nanoparticles, direct them towards a protein of interest, and quantitate levels of expression on those rare cells. So we've done that in a study of non-small cell lung cancer patients where we were profiling the protein PD-L1. So PD-L1 is of interest for cancer management and for therapeutic decision-making because it is a target of some of the immune checkpoints. So if you target PD-L1 on the surface of a tumor cell, that will affect the way the immune cell interacts with those tumor cells. And it's important to know whether the tumor is actually displaying PD-L1 to make a call as to whether or not a patient should receive immune checkpoint therapy. But that information can actually be difficult to get. A patient may have been biopsied many months or even many years before the decision is being made about therapy. And so what we were able to do was draw blood from these patients, measure the PD-L1 on their circulating tumor cells, which are very rare. Usually you get maybe 10, 20 in a milliliter of blood. And so through this quantitation that we did using magnetic ranking cytometry, we were actually able to pick out 3 different cohorts. So we sampled these patients right before they were given the checkpoint inhibitor. And then we were able to see that the patients who had disease that continued to progress actually had very high levels of PD-L1 on their circulating tumor cells. Patients that seemed to have stable disease when given the immune checkpoint had fairly low levels of PD-L1, but it was actually the moderate levels of PD-L1 that correlated with the patients having a complete response to the drug, which was kind of a surprising finding. We thought it would be high PD-L1, that those would be the patients that did the best, but that seems to be an indicator of disease that's kind of already spiraled out of control. And instead it's the moderate expressors that seem to be the best responders. So this provides another tool to give clinicians to allow them to select the right therapy for patients. We've also used a similar approach to look at not circulating tumor cells, but immune cells in the blood, very rare cells. And this was a collaboration with a thoracic surgeon in Toronto who runs a mesothelioma screening clinic and sees many patients every day that have been asbestos exposed. And the job there is to try to diagnose people with mesothelioma as early as possible so that the disease is more treatable. But there are not very good tools to be able to pick out those people that are developing these types of malignancies. And so we were able to hone in on a particular immune cell type that this thoracic surgeon kind of hypothesized could be something that could be used as a screening marker. We put the platform to work to find those rare immune cells in blood samples and found a really good signature that very clearly differentiated the asbestos exposed population from people that had developed mesothelioma. So this is another way that magnetic ranking cytometry can be used to put rare cell level information to work for the management of cancer. Another area where we have used this technology is in trying to do functional genomic studies with phenotypic readout. And let me explain what that means. So if you are going to embark on a CRISPR screen where you're going to take a guide RNA library, transfect it into cells, you then create a very large library of cells, about 1 billion cells. And then usually the end goal with these types of screens is to find a particular type of readout, to find a cell that's doing something special in that big collection, to provide information that gives you a way to link particular genotypes with the readout that you've selected. Most of the time, the readout is done on a live dead basis. So you put your guide libraries into cells, you have your collection of cells, you stress them, you subject them to some set of conditions, and then you look to see what cells lived and what cells died. And that can only give you so much information. What we thought would be extremely informative is to be able to read out this type of screen based on protein expression. And there had been a very limited number of studies in the literature where people had used FACS as a way to take a collection of cells and ask a question about protein expression. But again, with FACS, given the throughput, that was very challenging. So we thought with this ability to look at rare cells and characterize them and capture and sort them, that we might be able to come up with a way to really ramp up the throughput of this type of approach. So we did a proof of concept screen that was kind of our first foray into doing this. And the example that we chose to kind of launch this effort was a screen focused on CD47. So CD47 is a protein that tumor cells can overexpress to evade the immune system, kind of similar to PD-L1, but here the immune cell that they're trying to get away from are actually macrophages. So CD47 is expressed by tumor cells. The macrophages are coming along. They use a protein called SIRP-alpha. They kind of interrogate the surface of the tumor cell with SIRP-alpha. And if SIRP-alpha finds CD47, then the macrophage leaves the cell alone. So it's considered to be a don't eat me signal. So we wanted to ask the question with a phenotypic CRISPR screen, what genetic regulators might be there that would allow us to pull CD47 down, off the surface of the cell so that we could restore that interaction with immune cells and let them do their thing and take care of the tumor cells. So we went ahead and did that. We created our libraries of CRISPR edited cells. We used magnetic ranking cytometry to isolate subpopulations of cells according to expression levels. And we identified a really interesting hit in this screen, which is an enzyme called QPCTL. And this is an enzyme that actually cyclizes glutamine to pyroglutamate. And it wasn't completely clear what the link was in the beginning between QPCTL and CD47, but we eventually realized that there is a glutamine at the N terminus of CD47, and this enzyme was actually cyclizing it to form pyroglutamate. And so that was the actual phenotype that we were reading out with the screen, but we were quickly able to come up with a chemical inhibitor of QPCTL, this compound shown here that was actually looked at as a candidate molecule for Alzheimer's treatment. And with that small molecule in hand, we were able to show that we could inhibit the formation of that modification at the N terminus of CD47. And we're also able to show that that then takes down the binding with SIRP alpha. So you can disrupt that important interaction between tumor cells and macrophages by drugging this enzyme, which is a regulator of the production of functional CD47. And so with that, we were kind of on our way to discovering potential new therapeutics that could be used to go in and kind of get at that interaction between tumor cells and the immune system. And that's something that we're now continuing to pursue with a company called Evotec that's helping us with a very large high throughput screen to identify other chemical inhibitors of QPCTL. So this effort, this project or program where we're putting magnetic ranking cytometry to work to really drive the throughput of these types of phenotypic functional genomic screens is something that we're continuing to really build on and expand. And we think this is a very powerful approach for therapeutic target discovery and the eventual discovery of new therapies for disease. So the checkpoint inhibitor program focused on CD47 is kind of how we got going. We have a number of other checkpoint inhibitors that we're working with now. We've taken the same approach and used it to do in vivo screens. So there we're pulling rare cells out of animals that we've CRISPR edited and then can learn more about tumor progression that way. We have programs focused on undruggable proteins, things like KRAS, where we're looking at regulators of the expression of mutated forms of KRAS. We've built out the capability to look at secretion phenotypes. So instead of looking at expression, we can look at secretion. That allows us to look at the activity of T cells and to watch the levels of cytokines and other factors that are being secreted. We also have a really interesting collaboration with Amgen, where we're helping them build high performance secretion systems for therapeutic antibodies. So this is an area where we hope to kind of keep building and building and building based on this technology platform. So then what I want to do with the rest of the time that I have and make sure that we leave plenty of time for discussion is tell you about a new study that we just published that was our effort to take the same platform that I've described to you and to deploy it to advance a type of cell therapy called TIL therapy. And I'll tell you more about what TILs are in a moment. But this just came out a few weeks ago in Nature Biomedical Engineering. Got us a little bit of media attention that was very cool. Even got me a little snippet of time on NPR, which was very fun. But this is what I'd like to kind of spend the rest of the talk on. So the cell therapies that are making their way into the clinic, they come in a variety of flavors, but the ones that are making the speediest progress are based on T cells. So based on a type of immune cell that is responsible for doing active surveillance in the human body. So these cells are constantly looking for other cells that don't belong in the body and trying to eliminate them if they've determined that they're kind of a foreign species. And so this is obviously very important for clearing pathogens, being able to recognize a viral particle or a bacterial cell and then getting it out of the body. But it's also how our immune system works on clearing tumor cells. And sometimes we don't know how much of the time, but the immune system can clear a collection of tumor cells, a small tumor may be getting going. But some of the time, right, the tumors kind of grow out of control and then the immune system can't keep up. But just in terms of how T cells do their thing, there's two very important concepts. So how do T cells recognize cells that shouldn't be in the body? They do that using TCR. So this is the T cell receptor, and the T cell receptors will recognize factors on the surface of tumor cells and other cells. And that recognition event then causes them to secrete cytotoxic agents that will then kill the tumor cells. And so for a T cell to be effective here, it's got to have those two things. It's got to have a TCR that's going to recognize the cell, and then it's got to have the ability to secrete those cytotoxic molecules to eliminate the foreign cells. So then in terms of making this type of cell into a therapy, there are a variety of approaches that are out there. One of the ones that got to the clinic fastest is called CAR-T, where a chimeric antigen receptor has been engineered into T cells. And those are very effective against blood-based cancers, things like leukemia and lymphomas. And this type of approach, or all of the approaches that are shown here, these are referred to as autologous approaches. So here you take T cells out of a person. In the CAR-T example, you engineer in the CAR, and then you put the cells back into that same person. There are other approaches, which I'm not going to talk about today, that are referred to as allogeneic, where you have a kind of off-the-shelf cell type, maybe something that was derived from a stem cell. You can make very large lots of them. And that's also a very, very promising approach, but a little bit trickier, because when you take cells out of a person, engineer them a little bit, put them back in, the immune response is not going to be as dramatic as if you're taking an allogeneic cell type that maybe your body's never seen before. And so there are more safety issues potentially with allogeneic therapies. But that's probably, our future, where we do have off-the-shelf cell types. But until then, we have autologous approaches where we can use our own cells to potentially treat tumors. So there's the CAR approach. There's approaches where instead of installing a CAR, you can actually install a TCR that is going to be able to recognize a particular tumor antigen. That's been done and can be very powerful. But the approach I'm going to tell you about today actually looks at unengineered cells that are referred to as TILs. So TIL is short for tumor infiltrating lymphocyte. And these are cells, these are immune cells that infiltrate tumor tissue as a means to try to eliminate those foreign tumor cells. And I'll tell you a little bit more about how this works and how we've applied magnetic ranking cytometry to make this approach even potentially more powerful. So the way that TILs are produced at the moment is that tumor tissue is surgically resected from a patient. So you take a piece of tumor tissue, that piece of tissue gets shipped off to a manufacturing facility. Sometimes the tissue goes halfway across the country in order to get processed. It gets fragmented, it gets cultured, and during that culture step, what happens is that the cancer cells die and the lymphocytes grow up. And so you're left then with a bulk preparation of lymphocytes. Those go into a rapid expansion protocol where they're grown up to a very large cell number. Usually you need about 50 billion cells to infuse back into a patient for TIL therapy. Once the cells are at that level, they're cryopreserved, they're shipped back to the medical site and infused into patients along with a dose of IL-2 that helps the cells proliferate. So that's the way these cells are manufactured. And this is an approach that's actually been around for a long time. So it was first proposed by Steven Rosenberg at NCI in the 1980s. And at the time, it seemed like kind of a crazy idea to actually be taking cells out of people and putting them back into people. Now, we do all kinds of things with patients and patient treatment, and it's much more of a mainstream idea. And I'll talk a little bit more about this in a moment, but there have been incredible results achieved in the clinic with this type of therapy, with patients being cured of things like advanced metastatic melanoma. It's really the only cell therapy so far that is effective against solid tumors. So as I mentioned, the CAR-Ts are coming along, but mainly for things in the blood. TILs are actually effective against a variety of solid tumors. They're also very safe. So with CAR-Ts, you can have some adverse events. You can have cytokine storm that happens with some patients. TILs are very, very safe because these are just your own cells being put back into your body. Another thing that's very powerful about TILs is that you have a huge cocktail of TCRs in these TILs. They recognize many, many different tumor antigens. And so there's the ability to kind of chip away at many different clones within a tumor, which is important for evading resistance. So just to show you a little bit of the kinds of results that have been achieved in the clinic, a lot of the early work here is done in melanoma, where again, patients with if you look on the images on the left, I mean, this is some of the incredibly advanced melanoma, where 12 weeks after an infusion of TILs, all of their tumors were gone. And this response was durable. It lasted for years and years and years. Another study, this one's out of the NIH, where TILs were used to treat breast cancer. You can see on the left, this is with tumors present kind of in the breast, also liver metastases. After the TIL treatment, both types of tumors completely disappeared. And the images on the right were actually taken 5 years after the patient was treated with TILs. So again, a very, very durable response, a very long time after the cells were administered. So this is therapy where there's a lot of excitement about the promise here. But I think the thing that is that has tempered people's enthusiasm, it comes from a few different sources. One is that in the clinical studies of TIL therapy, the responses have been fairly uneven. So you do have some patients with these incredible responses where their disease just disappears, but you have some patients that don't respond, some patients who are in the middle. And so there isn't really a consistent result across large patient cohorts. Some other things that are issues are related to the manufacturing. I took you through the manufacturing process for doses of TIL therapy. It's a long process. It's expensive. The logistics are really complex. And the FDA also doesn't love the complexity of this. It's difficult to characterize what is actually in a dose of TILs. And so the regulatory approvals that companies like Iovance, which is a leader in this field, have been going after have taken a very, very long time. So there's lots to be done to make TILs more mainstream, to make them more effective. And one of the things that we started worrying about or thinking about was how to just make the doses of cells administered more consistent. Because if you think about what's in a dose of TILs, you're going to have many, many different T cell phenotypes in there. So T cells have this incredible ability to kind of morph between different phenotypes. In a piece of tumor tissue, you can have cells that are exhausted. So they've been in the tumor for quite a while. The exhaustion phenotype means that they can't really secrete anymore. They can't exert that cytotoxic activity. You're also going to have cells that are completely naive. So you have T cells that have just kind of come out of circulation. They got a little whiff of something interesting, and so they're starting to migrate towards the tumor. And they may have been kind of collecting at the periphery of the tumor when the tissue was excised, but they're not active. They're not tumor reactive. They don't have the right TCRs. And those cells in culture will expand very, very quickly because they're fresh, they're naive, they're not exhausted. And so they can very often overwhelm the more interesting clones that you might have within a T cell prep. And then in the sweet spot, you're going to have the kind of Goldilocks cells, the ones that are not too exhausted, but also not naive. They're going to be cytotoxic. They're going to be tumor reactive. So we thought using magnetic ranking cytometry, we should be able to find those cells and package those into a dose of TILs that then could maybe get around some of the heterogeneity that has been problematic here. And so the study that we did, and this effort was led by a very, very talented graduate student, Daniel Wang. And Daniel, one of the first things he had to do in order to flesh this out was to choose a marker, choose an antibody to put on our magnetic nanoparticles so that we could try to collect those Goldilocks cells in the sweet spot of reactivity. And so what Daniel chose based on what he could find in the literature is a protein called CD39. So CD39, it hydrolyzes ATP and ADP. It's important just in terms of regulating the tumor microenvironment. And it's also a marker that tends to have its expression increased as T cells are in the tumor microenvironment. So Daniel hypothesized that if we could isolate the TILs that had moderate levels of CD39, these would be cells that would be tumor reactive, but they wouldn't be exhausted. So he thought this would be a great place to start. So he went ahead and did that and isolated cells according to their CD39 levels. He did this using a mouse model of colon cancer. So he xenografted the tumors into mice. He then let the TILs infiltrate. He then resected the tumor tissue and isolated the TILs according to levels of expression of CD39. And what he found is that you have about 20% of the TILs that get extracted are in that subpopulation, so moderate levels of CD39. And then he went through and did a bunch of phenotyping, and this was really incredible to me that the phenotyping that he did was incredibly systematic. And when he looked at low CD39s, when he looked high, when he looked at the moderate expressors, that you could see that there was a big difference in stemness. So the mediums and the lows have a good stemness profile. They're not exhausted. They have a good proliferation profile, again, because they don't have the exhausted phenotype. And it was really the medium expressors that had the T cell memory phenotype, which is really what you want to hope to be able to get good persistence out of these cells. So he also did, that was some phenotyping that he did at the level of protein. He also did phenotyping at the level of mRNA and looked at markers of cytotoxicity. He looked at markers of stemness, of exhaustion. And again, he could see that same continuum of phenotypes for the highs, the mediums, and the lows. So we thought that these cells, these different subpopulations of cells really did seem to have properties that tracked with how we thought they might act and in terms of having anti-tumor activity. And so to test this out in vivo, he did a lot more in vitro that I won't talk about in the interest of time, but he was then able to take these cells that he isolated and sorted and generated those subpopulations. He expanded them to the levels that he needed to come into a mouse with a dose. So that's about 1 million cells, and injected these cells into mice and then looked at these different subpopulations and their ability to suppress tumor growth. So that's what he did in his in vivo study. And you can see here, so the black symbols are untreated mice. So those are the tumors getting larger and larger as a function of time in the mice that did not have any TILs infused. And then you can see that we've got the highs, we've got the lows. Those are suppressing tumor growth a little bit, but it's really the CD39 moderate cells that are doing the best job of keeping the cells in check. In this study, about 50% of the mice were actually cured of their tumors. They had no detectable tumors at the end of the study, so that was quite a dramatic result. And what we were able to show is that if you compare this subpopulation that Daniel isolated, and you compare that to what's being used in the clinic right now, that's these pink symbols. Those are unsorted TILs. That's the Iovance approach, where they grow all the lymphocytes out of the tumor tissue, package that into a cryopreserved dose, infuse it into patients. And you can see that there's a fairly small effect on tumor suppression, whereas when you use the subpopulation, you see a much more dramatic effect. So we think this is a really exciting way to think about maybe advancing TIL therapy, going in with subpopulations, something that's more consistent that we hope will allow a more consistent result to be achieved in the clinic. I'll just end with a couple words about kind of what Daniel's been working on recently. And this is very exciting because one of the big limitations in TIL therapy, I mean, there's all the things that I've talked about, manufacturing and consistency and all of these things, but the requirement for surgical resection is also a big limitation. You have to put a patient through surgery. You have to take a chunk of the tumor in order to get the access to TILs. And so Daniel wondered whether there might be any of these cells in the bloodstream. could we find tumor-reactive cells in the bloodstream and then eventually maybe be able to collect them non-invasively? That would be a game changer for this type of cell therapy. So he did a variety of studies, and I won't go deep into the biology here, but he basically set up a mouse model where he could track the TILs from one tumor to see if they showed up in another tumor, to ask the question, do TILs travel? So do they go back and forth between different tumor sites? And he was able to show that they do, that he could see TILs from one tumor ending up in another tumor. And so with that observation, that gave him some confidence that these cells were actually going through the bloodstream, traveling between different tumor sites. And so this got us, both very excited that maybe these cells could be collected non-invasively. And Daniel then went on to show that he could collect these tumor-reactive lymphocytes out of the circulation. So we call these CTRLs. And he was able to isolate these cells using our platform. They're very rare, so it's a very small number of cells that you end up collecting out of the blood. But it was enough for him to expand and to generate a dose that he could then study in a mouse model. And so here, this again is a colon cancer model. He's looking at, here's the untreated mice here on the left in the gray symbols. I'll just kind of go over to this data point in red where he's injected the CTRLs, and you can see there's a dramatic effect with the growth of the tumor being suppressed for a reasonable amount of time. He also combined this approach with an anti-PD-1 antibody, so with an immune checkpoint based approach. And the combination of the CTRLs and the anti-PD-1 antibody had a really dramatic effect and really just flatline the tumors. And you can see that the antibody on its own actually doesn't have an effect in this mouse model because it is immunocompromised. So you really need these external cells coming in for that to have any effect. And what was really neat about this study is that we were able to flatline these tumors to such an extent that most of the mice were cured. And so then we could keep those mice and allow them, or to keep studying them. And we could see that they remain tumor-free. And then Daniel actually did an experiment where he re-challenged these mice after 12 weeks to see if these cells were still around. Were they still active? Was he getting a durable response? And what he could see is that you could re-challenge them. You could come back in with another injection of tumor cells and kind of a second attempt at doing a xenograft. And maybe there's a little bit of a tumor taking off, but those cells that were injected weeks and weeks before are still around and are able to suppress the formation of a tumor. So this is something that we think is really exciting. Love to see this advance to the clinic. We have a new company that's called Control Therapeutics that is going to be trying to do just that, and would love to see this kind of non-invasive approach to autologous cell therapy make it to patients. So with that, I want to wrap up. I want to thank Daniel, who again is just a really incredible student who did all the work that I talked about in that last section of the talk. He's done many, many other things in my group that I don't have time to talk about. He's really a very just incredible scientist / engineer. I also want to mention Randy Atwall, who has moved from Toronto to Chicago with me. He's a research assistant professor out of biochemistry and molecular genetics, and he's kind of heading up our downtown lab. And then we have Mahmoud Labib, who also came from Toronto and is a research assistant professor in chemistry. And for those of you in Silverman Hall, you may have already met Mahmoud. He's based there and has really been instrumental in getting our lab off the ground in Silverman. So I want to thank all of them. There are many others to thank, everybody else in the group who's contributed to the stories that I told you today. Thanks so much again for having me and listening, and I'd be happy to answer your questions.
All right, thanks, Shane. I'm outside Silverman Hall. That was spectacular. And let the Questions fire, please.
Hi, Shana. This is Xiaoyu. Can you hear me?
Yes, I can hear you.
Great. So the TIL story is really cool. So have you ever thought about using this approach to treat like autoimmune disease? So in that case, you may like delete the T cells with median CD39 level.
Yeah, that's a great question. So we are looking at a lot of different versions of what I just told you about. Certainly there are other types of T cell based therapies that one could contemplate. Sometimes you really want to ramp up activity of T cells. Sometimes you want to take it down, right? And certainly being able to select subpopulations, learn how to deactivate these kinds of things we're also pursuing. So a lot of interesting directions to go, and we're kind of slowly but surely exploring the Northwestern ecosystem, and there's a lot of people around who have some really interesting ideas about T cell biology and how we might kind of work together to do that kind of thing.
Really cool. Yeah, look forward to more discoveries from your lab.
Thanks, Shayu.
Yeah, that's really interesting. I'd like to talk about cancer vaccines and how you might be able to meter out this. It's a very open-ended area. A lot of excitement, a lot of push from the Biden administration for Cancer Moonshot 2.0, and that's one element of it. So how might we approach using some of the things you talked about?
Yeah. Well, there's a lot of synergy actually between this type of therapy and the vaccine approaches. And that's something we've been exploring. If you look at a lot of the commercial players and some of the, oncolytic viruses, those kinds of things, there's a lot of combinations of those with TIL therapy. So I think, the vaccines can kind of get you so far, and then coming in with a selected cell type is a way to potentially augment. So we're looking at that as well.
Hi, this is Wen, and we're gonna CLP here. I find your magnetic ranking cytometry is very powerful. We work on the circulating tumor cells. We find usually tumor cells and lymphocyte cells, there usually be cluster. When the tumor cells cluster with the lymphocyte cells, usually they have poor prognosis and treatment. Can we use your technology to isolate those two cells together, that we can profiling, do all those kind of, analysis of why, because that's related to metastasis. Currently method we use cell search, and most of the time the cells are already fixed, right? We cannot recover them for culture or further analysis.
Yeah, that's a great question. So clusters are very interesting, right? So it's hard to see circulating tumor cells in the blood. It's really hard to see the clusters, but you can, and we've done it with this approach. And the clusters are so important, right? Because if you have cancer cells sticking together, they've been shown to have higher metastatic potential. And then if you have the lymphocytes in there too, there's a lot of information to be gleaned. So we haven't done clusters with lymphocytes, but we have done work on clusters in collaboration with a group in Italy, kind of taking our approach and then coupling it with a size-based profiling technology. Because the clusters are a little bit difficult for us to handle on their own, because if the size of the thing that we're isolating changes, that changes its drag profile, and then it kind of lands in a funny place on our device. But if we do size-based sorting and then we go into our chip, then we can get the same kind of information. So it's a really interesting problem.
Yeah, is that possible? We can use double antibody labeling with your mechanism? Because one antibody label CD35, another one like the tumor marker.
Yeah,
that sometimes you can pour those clusters there.
Yeah, that's an interesting idea. I mean, we can certainly use cocktails of antibodies, and we've done that for some other applications. So yeah, that can be done. And I've always thought it would be really interesting to isolate the clusters and then really look at the TCR tumor cell interactions that were giving you those clusters and to be able to characterize those kind of at the single cell level.
Yeah, I think this is a really fantastic approach to working on, because the current technology idea is, we cannot go further.
Yeah.
To do this study.
Yeah. Well, love to talk more with you about that.
All right, thank you.
Yeah, thanks.
Hi, thank you for your wonderful talk. I'm a grad student from the Chad Merkin Lab, and I have 2 questions. Oh, can you hear me?
Yes, I can hear you. Go ahead.
Oh, okay. All right. So my first question is, what's like the smallest change in mass that you can detect with this technique? And my other question is, what are some examples of changes in biological phenotypes that this can detect?
Yeah, great question. So I think your first one is about how much of a change can we detect? And here it would be in protein expression. So can we detect a 1% change or a 10% change, or does it have to be a really large change? And I we've done studies of that over the years. And I think, changes that are kind of 5 to 10%, those are pretty much at the edge of what we can resolve really, really well with very high precision. But that's usually good enough to be able to track most biological phenomena. And then in terms of what can we watch happening, we've done a lot of those types of studies. So we've looked at a process called EMT, the epithelial to mesenchymal transition. That's when cells lose their epithelialness, they become more mesenchymal. And that's very important for the metastasis of cancer. So you have a tumor, it's made of epithelial cells, but to leave the tumor, the cells have to kind of lose that property. So we've done studies monitoring that. We can look at proteins that are very involved in EMT and kind of watch those dynamics. That's been very interesting. We've done a lot of work, so I mentioned this at the beginning, looking at protein expression changes that may be relevant for choosing a therapy. Is a checkpoint molecule being overexpressed or underexpressed? So we've done some work there. And then a fairly new assay that we have that's very interesting is, instead of looking at expression, we can look at secretion. So I mentioned this, that one of the things that T cells do that can be very interesting to look at is they secrete cytotoxic molecules and other signaling molecules. And we've built assays that allow us to watch that secretion phenomenon. So that's been interesting too. But with this platform, what I love about it is that we can kind of keep building on it and building new types of analytical capabilities and new types of assays. So that's one of the things that we will continue doing now that we're here at Northwestern.
Yeah, it sounds really interesting. Thank you.
Yeah, very high modularity for sure. Are there maybe 1 or 2 more from the group?
Shana, I had a quick question about your circulating tumor-reactive lymphocytes. Since there's so few in the blood, have you thought about single cell TCR sequencing and doing like a TCR-T therapeutic? There are a lot of companies out there, because I've worked on T cell adoptive therapies, and it's great data in mice, but will you be able to get that scale, that manufacturing scale for human therapeutics? Because you're going to need hundreds of millions to billions probably, right? And I don't know where you are in terms of the GMP. Is your device GMP ready? Is your process ready for GMP and clinical grade TILs? So, but it's a really cool technology. I love it.
Yeah. Thank you, Terry. Yeah, I was going to say this is the $1 million question, but it's probably more like $100 million question, because what it takes to get these things out into the clinic. So, what's nice about our devices is that you can injection mold them. So we think the GMP compatibility is there. They're disposable, they're sterilizable. So that's all good. Are we going to be able to get enough cells? And for these circulating TRLs, that's the big question. Daniel's already done some work showing that he can find these cells in human specimens. So we've got that checkbox already done, but now we have to see if we can get to the levels. And the hope is that, you know, with the right selection approach, with maybe even a cocktail of markers, that we can push up to the level that you need for a dose. And this, you know, we're looking at a variety of ways to take this to the clinic. And this may end up being a little bit different from TILs. I mean, maybe it's not the therapy that you give to a patient with incredibly advanced disease, but maybe it's what you give to somebody who's just had a surgical resection of a large tumor, and you're just trying to kind of, you know, clear out any residual disease. And so there you might be able to work with a lower number of cells. So we're still contemplating that. And to me, you know, we've kind of done the heavy lifting here and shown that it works. And then the company will figure out, hopefully, how to get it to the clinic. But lots of different interesting possibilities there.
And I love the device, because one of the challenges for cell therapy, you know, like conventional drugs, you need to scale up. But this way you can scale out. And for autologous therapies, that's key. And I had a third question about what Neil was proposing for the tumor cells. I don't know whether you can, since you can sort them, you could maybe look for neoantigen specific mutations, either through sequencing or proteomics. I think that would be really cool if you could just get enough material to get the readout.
Yeah, absolutely. Well, Daniel's on it. You'll be happy to hear. And we're looking at all kinds of samples. I mean, the circulating tumor cells, you can look at those. They're pretty rare, so it's a little bit difficult. But we've been looking at pleural effusions and other types of samples where you can maybe get a few more cancer cells still. I mean, it's not that non-invasive, but it's better than taking a piece of tumor tissue. So there's other ways to get at that, and we're working on that quite actively.
Great.
Yeah.
I'd love to talk about it.
Thanks, Terry. Yeah, I totally agree. The scalability in getting to the cell counts for proteomics, even as it approaches towards a few thousand cells, ability to do the kind of experiment you just said. But Shana could get 10,000 or 100,000 cells and makes it feasible to do what you just said. So super exciting all the way around. We had one straight question in the chat, but I think that person left. So why don't we call it good there and release Shana into her Thursday. This has been CLP Chalk Talk, and is so infused with such great technology, bioanalytical, chemistry, engineering at the intersection of cancer biology and all those disciplines. So truly transdisciplinary. I'm Neil, your host, and I double as a weatherman, and I happen to be outside. So I can tell you that if you're going home in about an hour, you're going to want to bring your umbrella. Thank you, Shana. Thank you to everybody. And we'll see you next time. Have a good night.
Bye.
Thank you.